Well, thank you everyone for joining us today. So I'm basically talking to you at 7 p.m. here in Australia. I'm gonna talk about a new development that we did, which is our low-cost EDU multiplexing methodology for understanding and for studying the invocation and cell cycle dynamics. And to get us started, basically, we're interested in the cell cycle, which is a fundamental process in cell division and plays a crucial role in various cellular functions such as cell commitment, tissue formation and development, and it also maintains homeostasis in a variety of different tissues. We want to understand its core phases and regulatory mechanisms because dysregulation of it can lead to congenital malformations, as well as it's very well known for its sort of controlling role in cancer. The cell cycle consists, as you can see here in this slide on the left, of four major discrete phases, starting by the phase of GAF1 or G1, followed by S phase of denatrification, G2, and later on mitosis. And we have been using, or researchers employed a variety of different tools and strategies to map the progression of it and its phases, such as markers including K67, phosphohistone 3, PCNA or Aurora B, and do also cycle reporters like Fuji and DNA dyes used to assess DNA content. Detection methods for denatrification or synthesis have traditionally utilized analogs of thymidine that can be readily incorporated into replicating DNA. An analysis of these tag cells, as you can see here, can later on allows for the identification of denatrification events, providing information of the rate of synthesis, extent, and also the genomic location. And different approaches have been employed over time, starting with radiation detection of thymidine around the 50s, the antibody detection of VRDU from the 80s, and followed by most recently the click chemistry based assessment of EDU uptake, known as click chemistry. EDU here, as you can see in this schematic, is detected using click chemistry, which is a revolutionary technique allowing for the fast and efficient conjugation of molecular entities, which was in 2022 recognized with the Nobel Prize in Chemistry. Remarkably, EDU has several advantages over BRDU, and one of the most significant ones I would say, and I wanted to highlight it today, is the alkyne here is not affected by any steric hindrance of the DNA, so you don't need those sort of harsh DNA denaturation protocols. And as you can see, and I guess this might be a consequence of it, EDU somehow has been replacing the use of BRDU. This is just a PubMed search that we recently published, actually, as part of the manuscript that I'm going to be showing you today. And up to now, we have about 3,000 total studies utilizing EDU itself. So since 2008, EDU has been widely utilized for the analysis of denatrification, cell proliferation, and differentiation as well, and also for the identification of different phases of the cell cycle when combined, for example, in this case, by flow cytometry with DNA content assessment. EDU has also been used for high resolution microscopy of the application sites here on the left for chromosomal labeling, as well as to understand root plant growth. EDU has revealed the proliferative nature of the stem cell gut niche, and I'm going to be showing some images today about the same neurogenesis, and more recently, a paper in development used it for the evaluation of chylomyocyte DNA synthesis after birth. So something that I wanted to highlight today is that despite EDU being used as a tool for standard application, it's mostly sold as a kit. And of course, kits have several disadvantages, and I'm just showing you here a few or a bunch of them, such as unknown reagents in general, not allowing for in-house optimization. They're expensive. They sell normally kits for imaging or flow cytometry. They're not really that user-friendly. They're normally limited in colors, 3 to 5 colors depending on the assay, fluoroassay conjugated options, and they have low performance in 3D organs and tissues. So to address these issues, we recently developed a new strategy known as OpenEMMU, as I mentioned this morning. And here is just some of the cover proposal we did to the journal. You can find the full story here published just a few weeks ago. And this stands for being a versatile and open source EDU multiplexing methodology for understanding DNA and cell cycle. So to begin with, what we did at the beginning was to optimize every single reagent that you kind of need for the click chemistry based copper-based detection of EDU. And this is just an example how we optimize the copper sulfate concentration. We have been working around 800 micromolar concentration for copper sulfate because that was one of the ones that gave us better discrimination of EDU positive cells. Something that we have been also looking into it is the optimization of other reagents such as the reductant to perform this kind of a strategy, and we have been using ascorbic acid. Something that we also put together was the use of this sort of ASI dyed 488, and we use actually a variety of different colors, and we selected 200 nanomolar of the right concentration of the azide to use. Something to highlight actually about this point is that not because we increase the concentration of the azide, which is kind of what the kit does in general, you actually have better performance, but it's the other way around. We have also worked around different serums, and people mostly use FBS, but it's more expensive in general than NCS. And we demonstrated that no matter the serum that you use, our protocol sort of excels, as well as no matter what DNA dye you use, we can use a different variety of DNA dyes, and they all sort of perform well in terms of allowing DNA content discrimination together with EDU to evaluate DNA synthesis. So this obviously demonstrates the capabilities that we have created using in-house reagents and using a click chemistry-based reaction. We can generate labeling of replicative DNA and cell cycle progression using OpenEMMU, and this is just a demonstration of how using, for example, a quick labeling of 1 hour of these are human-induced polymerase stem cells, how we can separate cells in the different stages of the cell cycle, such as G1, LES, mid-S, late-S, and G2 cells, as well as mitotic cells that are EDU, obviously negative. And this also tells us about the DNA behavior and the labeling of EDU in this case. Something that we as a team are very proud of is that we're not only having a superior in-house and low-cost strategies, that we actually evaluated the cost of what we generated name as OpenEMMU, and we compare that with a variety of different kits. And I'm just showing you here two mostly sold around. One is Vector Labs, which is kit number 1 here, and the other one is the Invitogen kit here, as you can see. And we calculated the cost of every reagent. And in about 2 years that it more or less took us to put together this, we basically save about 17, 18 thousand pounds, or about 20,000 US dollars, which is an equivalent, at least in Australia, of 180 RNA sequencing samples, or a reasonable big size single cell RNA sequencing dataset or experiment. So this obviously comes with a lot of savings, as you can imagine. We have thoroughly evaluated our methodology using several immortalized cell lines, and this is just a few to name and some examples here, but such as fibroblasts, calves, and muscle stem cells, mouse and human cell lines, also immortalized ones, and iPSCs and iPSC-derived cells, as well as immune cells. And I'm going to be just showing you some examples about the work that we have developed. So in addition to its compatibility, as I just showed you with flow cytometry, for example, we have also developed at the same time imaging and automated segmentation and analysis. And something that we also wanted to evaluate is whether our resource or toolkit surpasses the Invitogen kit in terms of de-amplification and cell separation. And that was a key element, because we not only wanted to create something that it was low cost and reproducible, but we also wanted to demonstrate that we can create something superior to what is available in the market. And this is just an example how, we can clearly demonstrate that OpenEMMU enables a better separation of DNA replicating. Cardiomyocyte, these are cardiomyocyte at day 20 of differentiation. These are separated by cardiac 1 and T, positive levels. And here you can see how, OpenEMMU is basically 10 times brighter than the Invitogen kit. This is in the log scale. We also demonstrated that this not only performs well in vitro or using in vitro cells, but we can also inject EDU into mouse models and evaluate the proliferation of endogenous cells, such as in this case, you can see here bone marrow cells. So these cells were isolated, and we perform again the click chemistry side-by-side comparisons using the same cells for OpenEMMU and the Invitogen kit, and again supporting that our OpenEMMU outperforms Invitogen or any commercial kit available to date. We also compare the antibody and multiplexing compatibility of this click chemistry in-house that we develop, observing that OpenEMMU outperforms Invitogen when we were combining the detection of EDU now with the detection of, for example, the phosphorylation of histone 710 for the detection of mitotic cells in the bone marrow cells. And as you can see here, we're not only observing the true mitotic cells after the click chemistry, but we can also detect more cells, and the labeling being actually way stronger, which obviously facilitates the study of mitotic cells in such case. We have further developed into better understanding of how we can multiplex, in flow cytometry. And I'm going to show you some images about multiplexing in microscopy. And this is just a bit of the same story. Basically, we use human T cells, our PBMC assay. We generated some sort of different challenges from spike, SARS-CoV-2, some bacteria and influenza, and classical T cell challenges, such as this kind of activation driven by ligands. And as you can see here, we can see CD25 positive cells, which are differentiated T cells, but also those, or a large variety or majority of them being also positive for EDU, demonstrating again the compatibility of what we have created with multiplexing strategies and adding more colors on top of the detection of EDU already and DNA as well. So OpenEMMU not only enables classical tissue sections or paraffin or sections, but it also allows for 3D biology. So in this case, we have combined the detection of EDU with ethyl cyanamide tissue clearing strategies, and those images or results can be seen here at the top. Something that kind of call our attention and is kind of, we mentioned this in the manuscript, is that it's basically K67, that is general marker of proliferation, seemed not to be a very good one, because when we did a very short pulse of EDU, not every EDU cell came positive for K67, and about 1/3 of all the cells that we could detect as EDU positive. And as you can see here, I've highlighted some of these cells with these lines here. They actually happen to be mostly negative for K67, even when we know that those cells are actively proliferating, because those are the transit amplifying cells in the gut that labels the stem cell niche. So obviously this is highly relevant, and as you can imagine, lots of people normally use K67, and we're trying to move forward the idea of people should not necessarily just using K67 when they study cell cycle and proliferation in their very own studies. We have also done, this at a large scale. So this is a tiling reconstruction of about 250 images here on the left. We are obviously very interested in understanding embryonic development, and a lot of the body that I'm developing right now stems from that kind of work. And here you can distinguish distinct tissues and organs that develop and grow at different rates. And we have observed likely highly proliferative ones, including the liver, the brain, and some regions of the brain and the lung. But we have also seen in the same embryo, using the same strategy, other tissues that grow at a limiting pace, such as the heart and some regions of the kidney. So OpenEMMU is obviously now allowing us in this amplifying new space to explore spatial biology and cell cycle activity using low-cost strategies. As I mentioned at the beginning, we're really interested in also validating strategies for multiplexing. This is just an example of what we have been able to do, mixing now OpenEMMU with IBEX. That is another strategy to do bleaching and recapturing of a secondary antibody, again allowing for a variety of cycles of staining. And here's just an example how we have been doing 12 or plus colors coming from a single slide and a single image. We have done this also in 3D using now deep clear strategies or other strategies that are not necessarily cyanamide compatible, but other sources. And here's just how we have now been profiling heart development at the 3D level. We can do imaging and microscopy of a depth of more than 1 millimeter in depth. And you can also see how we can, observe with detail and single cell resolution. And we are now developing this not only in the heart, but also in challenging tissues such as the growing limb, identifying key morphological structures such as muscle groups and other structures, fine structures such as the fingers. We have also provided detailed maps of actively proliferating regions, and I'm about to finish now using other classical models of development such as zebrafish. And this is just a FFPE section and DeepRFNIs that we've done with OpenEMMU as well, allowing to detect different actively proliferating regions across the growing larva. But we've done this not only in sections. We have obviously developed now using deep clear tissue strategies, developed 3D models of active proliferating regions across the larva and passing by, for example, the caudal fin, as you can see here, the fin musculature, the pectoral fin, the brain, and different sections of the head, including the eye and the retina, that are known for being highly proliferative regions in the growing larva, including. So to finish today, I just wanted to explain a little bit how we have recently solved a bit of a developmental problem, And we have recently demonstrated that global deunification stress impairs cell fission baryogenesis. And the way that we did this was by following a Nature Cell biology paper where the authors recently used acetylcholine in combination with hydroxyurea in order to stop DNA replication. We've done a single now combination of those two using acetylcholine, that is a non-DNA polymerase beta class inhibitor. And we have observed that acetylcholine treated embryos exhibited developmental abnormalities, including delay eye and head morphogenesis and fragility and outer body axis elongation, as you can see in some of these images. And finally, what we really did was to do a very short pulse of VDU at the very end of this treatment, from 25 hours post this sort of, or 20 hours so part of the challenging. And as you can see, deunification stress led to a pronounced reduction of DNA replication labeled here by EDU, but also we observe a reduced hematotic activity labeled by phosphohistone 3. So to summarize, a lot of the work that we recently put in a mouse script, we have basically addressed several issues. We're now working with known reagents. OpenEMMU is totally inexpensive, and it allows for continuing developing long strategies and long-standing, and to resolve long-standing questions in cell cycle, in the cell cycle field. And thank you for everyone for listening today, and thank you, Alex, for the invite, and obviously the team behind and all my funding sources and sponsors.
Wonderful. Thank you very much, Osvaldo. A reminder to the attendees to please ask your questions using the Q&A box. And while you're typing those out, I can start with a couple of questions. So you showed loads of beautiful images in lots of different systems. To what extent are you using a standard protocol to generate all those images compared to needing some optimization for the tissue or the structure that you're looking at?
Um, that's a great question, and we actually provided in the manuscript, and it was a bit of an issue with the editorial in general, because we put a very long protocols, and something that I can mention, we have been now in contact with many people around the world asking us for our protocols, and they have been able to actually reproduce a lot of what we've done just from the bioRxiv version of our paper. And that was amazing. And I got someone who was working skeletal muscle, 3D reconstruction after muscle injury because the muscle stem cells exist, PAX7 positive cells. And they have been able to reproduce a lot of what we've done in-house just buying the reagents that we declared. So actually not a lot of optimization. So it's pretty reusable as well. And that's something that we're obviously very happy to announce, and we will be very happy to share protocols in case people need them.
Great, thank you. And my other question is thinking about the limitations. Are there any is there any areas that you're aware of that struggle to be captured by this tool at the moment? So, for example, if you're looking in vivo at hematopoietic stem cells that are embedded in bone marrow, do you have a penetration issue in getting EDU into those cells? Are there any things that you're aware of that don't work quite as well as you'd like to at the moment?
Um, yes and no, and actually that was one of the reasons why we started doing this. So we saw some deficiencies in labeling when using some challenges tissues, as you're kind of mentioning. And we actually observed that that was actually given because of the kit and not having a full-on optimized. And so actually, when you think about 10 times beta, or a reduction of 10 times in, efficiency, it's not that just turn on the power in microscopy. Um, and we have solved a lot of those sort of penetration and or intensity issues by now having a much better and more optimized strategy. So yes, some tissues are more challenging to work with, but I think this is probably solving a lot of those issues. And actually, that was one of the motivations that drove this work initially.
Sure, great, thank you.